Hello everyone, welcome to the ITLS Academy. Today we are going to discuss another topic in the series of food preservation, that is your food spoilage. So simply food spoilage, if we talk about the meaning of that food spoilage, so simply it is what? The quality of the food product degraded. Now, the quality means that either the chemical composition of the food product, chemical composition of the food product degraded, or either the physical or sensory quality of the food product degraded. Either the physical or sensory quality of the food product degraded. That is known as a food spoilage. Means that when if we consume that type of the food product, then it is very harmful for us. Okay, so these food spoilage is affected by any chemical or sometime the microorganism, especially by the bacterial effect. Okay, suppose that if we keep the food product for 2 hours at the temperature range at between like suppose that the 37 degrees Celsius. Now due to that high temperature or longer period of the time, the microbial growth occur on that food product. Okay, now we can say that at that time the food are spoiled. So this spoilage are what? Their quality of the food product are degraded. If the spoiled food are consumed by the consumer, then it is very harmful for them. So let's start today's lecture, that is your food spoilage. What is spoilage? We all know that this spoilage is what? It is the either the chemical composition, physical appearance, or the sensory quality of the food product are degraded. Okay, so that food product are not in a good characteristic as long as, and it is not acceptance for the consumption of the consumer in a longer way, then that is known as a food spoilage. When a food is spoiled, its characteristic are changed. Okay, the characteristic that I already told you about, that either the chemical characteristic, physical characteristic, or sensory quality. If there are changes, then that means that it is not acceptable for the longer period of the time. Okay, spoilage is a subjective quality. What is spoiled for one person may be perfectly acceptable to the another one. Sometimes the food are what? If the food are spoiled, sometime it is accepted by the one person. Okay, means that their immune system are stronger and they can consume. If they consume that type of the food product, then there is no effect on their body. But if somebody whose immune system is very weak, and if they consume that particular type of the food product, then it is affected to that person surely. If microbial product associated with the spoilage X of order has certain detection beginning. Okay, suppose that if a food is spoiled by the microorganisms, okay, that spoilage are also take place by different method, either the chemical method, either the physical method, and sometimes the allergen, and also sometimes the microbial uh, like microbial production on that food product. Okay, so if the food are spoiled by the microbial production, then X of order has certain the detection of the beginning. The level will be below this beginning for most of the product are acceptable shelf life, Once it reaches, it will repeatedly passes so that in a short time after the level will be excess of that beginning and the product will be profoundly spoilage. Next is your what is spoilage. Okay, so that is the changes occur when a food when the changes occur in a food product and that changes are not acceptable, then that is known as a spoilage. Okay, so these changes are occur either by the microbial activity into the food product and sometimes the non microbial activity. Okay, so if the food are spoiled by the non microbial origin, then the result is insect damage, drying out, discoloration, staling, rancidity. And if the food is spoiled by the microbial activity, okay, this is a most common type of the cases in which the food are spoiled by the microbial activity. So microbial growth in the form of surface of slime or colonies. Sometimes the microbial growth are occur into the food product either in a slime or either in a colonies. Degradation of the structural component of the food which causes the loss of the texture. Chemical product of the microbial metabolism, gas, pigments, polysaccharide, off odors and flavor, they are the most common type of the degradation occur due to that microbial activity. Like it can affect the metabolism, it can produce the gas, it can affect the pigments, polysaccharide, off odor and flavors. What is spoilage? So it is a graph between the time and the count in millions. You can say that when the timer increases, the number of the bacteria are increases. Okay, you can say that this line is marked as a detection beginning, means that here the food spoilage are occur, or you can say that the food spoilage are started. So population level to produce detectable off order and product acceptable shelf life. Okay, so this is about the graph which shows that growth of spoilage is plotted on a linear scale. Next is your, so the first food that we discuss here is a milk. Okay, so we all know that milk is a what? Milk is a fluid which we excluded from the colostrum. It is secreted by the mammals for the nourishment of their young one. Okay, and colostrum is what? Colostrum is a more concentrated form of the liquid which contain around 25% of the solid, and in colostrum mainly the proteins are present. Okay, and it is secreted immediately after the parturition. The principal component of the milk are what? Milk are composed of the water, fat, protein and lactose. These are what? These are the principal component for the milk. Okay, and the precise composition are varies but between the species. Okay, the amount of these ingredients which is present into the food present into the milk are totally differ, and it is depend upon the species. There can be compositional difference between the breeds of a single species even within a single breeds. Typically the composition of the milk are like variation of the composition are depend upon the factor like stage of the lactation, okay, milking, and interval between the milking and the time of the day, the number of the previous lactation, and the nutritional and health of the cow. Their composition are depend upon all these factors, that is your stage of the lactation, stage of the milking, interval between the milking, time of the day in which the milking are performed, and the number of the previous lactation, and generally the nutritional value of the cow and the health of the cow. Lipid content is the most variable feature. It contains approximately 80 to 85% of the protein, which is your caseins are present. Milk specific protein are precipitated by decreasing their pH. So this is a composition of the whole milk. Like the water content is around 87.1%, fat content is around 3.9%, protein is around 3.4%, lactose is around 4.8%, minerals is around 0.8%, and total is about 100%. When we have to decrease some specific type of the protein, okay, suppose that when we have to precipitate that milk protein, then what we have to do, we decrease the pH of the milk, then it reaches to the 4.6. It's high water activity, moderate pH. We all know that the water concentration of the milk is high, and the moderate concentration, that is around 6.4 to 6.6, and supply the nutrient of the milk like excellent medium for the microbial growth. Okay, all the factors like your moderate pH, high water activity, like that means that the high moisture content, and high nutritive value, these are all these are what suitable for the microbial growth. Milk dirt poses a number of the antimicrobial features which protect the udder from the infection and newborn calf. Okay, we all know that microbial growth are very higher in case of the milk, but there are this milk are poses a number of the antimicrobial features which protect the udder from the infection and newborn calf. Generally, these are present to low concentration in cow milk to have a very marked effect on its keeping quality or the safety. In some cases, the antimicrobial activities is antagonized by other milk constitute like your effect of the citrate and bicarbonate on the lactoferrin activity. Three sources which contribute the microorganisms which are present into the milk. The first one is your udder interior. If the udder interior are not properly clean, then it also contribute the microorganisms sources into the milk. That exterior and its immediate surrounding environment is very important. We all know that when the milking are done, okay, within like 5 to 10 seconds, the milk are um, free from any type of the microbial effect. But when the time is increases, the microbial load are also increases. So these microbial load are increases either due to their surrounding, either due to the milking process, either due to the milking handling equipments like udder interior and feet interior. They all are supported the growth of the microorganism into the milk. Bacteria that get into the outside of the tet may be able to invent the opening and then the udder inferior. Aseptically taken milk from healthy cow normally contain around low number of the organisms. Okay, if we perform a milking in a aseptically condition, like that the environment which are free from any type of the microorganism, then it contributed a low number of the microorganisms growth into the cow milk. Fewer than 10 to the power minus 2 to 10 to the power minus 3 CFU per ml, that is colony forming unit. So around 10 to the power minus 2 to 10 to the power minus 3 CFU per ml bacterial load are present into the cow milk. And milk drawn from some quarter by the sterile condition. The organisms not commonly isolate are micrococci, streptococci, and corynebacterium bovis. Counts are higher due to the mastitis and inflammatory disease of the mammalian tissue. This mastitis is what? It is a inflammatory disease which occur in a mammalian tissues, due to which the microbial load into the milk are higher, and that disease is known as a mastitis, which is a major cause of economic loss in a dairy industry. So due to this, when the higher microbial load into the milk occurred, then the major cause of economic occur in a dairy industry. In the early acute stage of illness, the bacterial count in mastitic milk can exceed around 10 to the power minus 8 CFU per ml, and microscopic changes are often visible in the milk. These mastitis are diagnosed by the presence of higher number of granulocytes that can rises to 10 to the power minus 7 per ml. Okay, when we have to diagnose the mastitis, then it can be diagnosed either by the presence of the high number of the granulites which are present into the milk, and it reaches to the around 10 to the power minus 7 per ml. The most important organisms which occur due to that mastitis are Staphylococcus aureus, E. coli, Streptococcus agalactia. These are the three potential human pathogens. Okay, we all know that if these three pathogens present into the food products, then it can affect the health of the human being. That is your Staphylococcus aureus, E. coli, and Streptococcus agalactia. They are present into the milk by milk when a mastitis stage are occur. The next is your Streptococcus eubareus, Pseudomonas aeruginosa, and Corynebacterium pyogenes, and the number of other human pathogens like your Salmonella, Listeria, monocytogenes, Mycobacterium bovis, and Mycobacterium tuberculosis. Infected cow are then treated by the antibiotic injection into the udder. Milk from these cow must be withheld from the sale of several days because these antibiotic residue can cause up problems in sensitive consumer and inhibit starter culture activity in a fermented milk. Okay, so when a cow are infected, then the cow get a injection of the antibiotics into the udder. Okay, and due to that antibiotics injection, the cows are the sale for the sale of the milk of that type of the cow are withheld. Okay, because when the antibiotic residue are present, then maybe these the residue of the antibiotic can sensitive for the consumer, and it also inhibit the starter culture activity for the fermented milk product, because we all know that when we have to produce a fermented milk, then we required a starter culture, but due to that antibiotic effect, the starter culture activity also inhibited. Attempt to control the mastitis by good milking hygiene and use of disinfected tea dip after milking and antibiotic fusion at the end of the lactation have helped to reduce that streptococcal and staphylococcal infection, but have a little success in prevention of that E. coli mastitis. Okay, so these mastitis can be controlled by good milking hygiene process. Okay, and it can be also disinfected when we treat our milking with the antibiotic infusion. That is it can reduces the streptococcal and staphylococcus infection, but we get a very little success in case of the preventing the E. coli mastitis. The other exterior and its immediate environment can be contaminated with the organisms from the cow general environment. This is less in the summer when cow allowed to graze in open pasture, and worst when they are housed indoor under the wet condition. Heavily contaminated treats also contributed around 10 to the power 5 CFU per ml in the milk. Contaminating from bedding and manure can also be the source of the human pathogens, that is your E. coli, Campylobacter, Salmonella, and Bacillus species. It can also introduce by the soil to the milk. Clostridia can get into the milk from the silage, and their growth can cause the problem that is known as a late blowing in some cheeses. Milk handling equipments like your teat, cups, pipework, milk holders and storage tank, they are also have a principal source of the growth of the microorganisms. That's why we have to sterile the milk component also. Suppose that the milking are done in a proper hygienic condition, okay, but the equipment in which we do our milking, it is not a hygiene. So like and the equipment, that is your teat cups, pipe work, and milk holder and storage tank. Now what you do, you all follow all the protocols which prevent the microbial growth, but due to the milking handling equipments, even the milkman can contribute the contamination into the milk. So this is very important to maintain the hygiene in all level. This is a primary principal source of the microorganisms which are present into the raw milk. Milk is nutritional medium, and if equipment is poorly cleaned, milk residue on a surface that are frequently left wet will act as a focus for the microbial growth, which can be contaminated subsequent batches of the milk. Okay, we all know that this milk is what? Milk is a nutritional medium. Okay, and nutritional medium means that it is a good source for the growth of the microorganism. When the milk are not properly clean, or when the milk equipments are not properly clean, then it can um, affect the quality of the food product, and and like grow the microbial load into the milk product. To encourage the farmer to apply the available advice, the Milk Marketing Board, that is MMB, in England and Wales introduced a system of paying farmer based upon the total bacterial count of their milk. If the total bacterial count is high, then they get a less amount for their milk. Okay. In most developed countries, milk is chilled immediately after it issue from the cow. Okay, so there are developed. If we talk about the developed country, then what they do when when the milking is done, they immediately chill that milk after its tissue from the cow, and it held at low temperature, stored in refrigerated holding tent, then transported by the refrigerated lorry to the dairy. Keep in chill storage tank until it is used. Okay, so the first thing is that what we have to do, we keep a milk in a very chilling condition. Okay, either we store it, either we transport it, and either we again do a processing. So it will all started at a lower temperature. If the temperature below a 7 degrees Celsius, the only organism are capable of growing on the milk is psychotroph, because can grow at a lower temperature. The most commonly psychotrophic species present into the milk, like your gram-negative rod of genera, Pseudomonas, Acinetobacter, Flavobacterium, and if we talk about the gram-positive bacteria, then the Bacillus species can grow under the below 7 degrees Celsius. Okay, so after the milk, the second food product, that is your meat, which is also a good source of the microorganism due to the high concentration of the nutritive value. Okay, originally this meat was a term used to describe any solid food. It has played a significant role in human diet since the days of hunting and gathering. Okay, we all know that this meat are what it is play a significant role in a human diet, because it give a taste, and along with the taste it also give a nutritive value to the milk. That's why the human being take it in their diet. But if we talk about the initial time, then in that time the milk term are only used to describe the solid food product. Mean Meat eating remain widely popular today. This is due to its desirable texture and flavor characteristic that I already told you. That nowadays the consumption of the meat is high. Why it is high? Because their desirable texture and flavor characteristic, and also due to the nutritive value of the milk. Value of the meat. Although these meat protein does not does also have a high biological value. Numerous species are used as a source of meat around the world. The meat animal of principal importance in economic term, that is your cattle, sheep, goats, and poultry. Edible animal flesh comprises principally the muscular tissues. Okay, the edible part of the meat is what? It is the muscular tissue, but also include the organs such as your heart, liver, and kidney. Most microbiological studies on a meat have been conducted with the muscular tissues, and it is on these that the information presented here is based. How in many respects the microbiological will be broadly similar for other tissues, it should be remembered that the differences may arise from the particular aspect of their composition and microflora. Okay, so basically the microbiological study of the meat are totally depend upon the muscular tissues of the meat, because these muscular tissues are consumed in a higher demand. And apart from the this muscular tissues of the animal, other organs like your heart, liver, and kidney are also consumed by the human being. Okay, next is your, it is we all know that meat have what? Meat have higher water content, like approx 85% of the moisture present into the meat. Moisture present into the meat. Okay, and also have high nutritive value. Due to that, higher water content and nutritive value, the meat become an excellent source for the microbial growth. Both type, that is your proteolytic, the carbohydrate content of the muscles, and has a partial important role on its microbiology. After the death of the glycolytic breakdown of the glycogen continue leading to an accumulation of the lactic acid, which is decrease in muscles pH. When the death of the animal occur, the glycolytic breakdown occur, and it, due to that glycolytic breakdown, glycogen continuous leads to the accumulation of the lactic acid. Glycogen are produced, and this glycogen leads to the accumulation of the lactic acid, and these accumulation of the lactic acid decreases the pH of the muscles of the meat. In a typical mammalian muscle, the pH will be dropped around 7 to 5.4 to 5.5 with the accumulation of the lactic acid. Now, pH is less, the nutritive value of the meat is high, and the water content is high. All these are support the growth of the microorganism. If the muscles have been exercised before the slaughter, we will help a higher ultimate pH. The higher pH will also mean that microbial growth is faster, so the spoilage will occur sooner. When the when the pH is high, then the microbial growth is faster and spoilage will occur sooner. The microbiology primary processing. The tissue of a healthy animal are protected against the infection by combining of the physical barrier and the activity of the immune system. Meat from the freshly slaughtered animal should be relatively free from any type of the microorganisms. Okay, when meat are slaughtered freshly, in that time the meat are free from any type of the microorganism, but microbial load are increases under the stress, and they will be course higher if the meat the animal are suffering from any type of the infection. Or when if our milk are fresh, then microbial load are less. But when a timer passes, the microbial load are increases on that freshly prepared meat. But microbial load is increases under a certain strict condition, if animal are suffering from any type of the infection, then obviously the microbial load are higher during the freshly slaughtered milk also. The most heavily colonies area of the animals that contaminated meat are skin and gastrointestinal tract. Okay, so these skin and the gastrointestinal tract both are what? Both have a heavily colonized area of the milk for the contamination. Number and type of the organisms are carried at these sites will reflect both the animal indigenous flora and its environment. For example, the animal carry a mixed microbial population like your micrococci, staphylococcus, pseudomonas, yeast and mold, as well as microorganism which are derived from the fecus and the soil. They are what they contributed the contamination into the meat. The greatest opportunity for the contamination from the animal themselves occur during the dressing, the stages during which the head, feet, hide, and excess fat, viscera, and offal are separated from the bone and muscular tissues. Skinning can separate the contamination from the hide to the freshly exposed surface of the carcass through the direct contact and via the skinning knife or handling. After dressing, meat is cooled. When the dressing is done, then meat is cooled to chill the temperature, during which cold shock may cause some reduction in a number. Okay, maybe when a cold shock occurs, then it reduces the contamination in it reduces the number of the microorganism present into the milk meat. At chill temperature, microbial growth is restricted to the psychrotrophs, and these can be further inhibited by partially surface drying than take place. Okay, when the meat are stored at a low temperature, then there is a chances of the growth of the psychrotroph which can grow at a low temperature. Okay, but it can be inhibited when a partial surface drying are occur. Then we can inhibited the psychotrophs also. Counts are generally higher in case of sheep than a beef and higher still in a pigs. In primary processing of the poultry during the transport to the plant, contamination can spread between the birds by fecus, feather, and from poorly cleaned transport cages. The intestinal tract of poultry will contain a higher number of the microorganisms, and the pathogens that present into the intestinal tracts are Salmonella and Campylobacter. Poultry to be frozen is usually chilled in water, and this offers a further opportunity for a cross contamination. Aerobic storage of chilled red meat. Now we are going to discuss about the spoilage of the fresh milk. Okay, now we all know that when a aerobic storage occur in case of the chilled red milk, so either it occur at unwrapped milk meat or are covered with a oxygen permeable film. We all know that when we kept our meat in a chilling condition. Okay, so there is aerobic storage reaches, so it reaches either when we keep the meat at unwrapped condition or we covered the meat with a oxygen permeable film. It produces a high potential at the meat surface which are suitable for the growth of the psychotrophic aerobes. Non-fermentative gram-negative rods grow most rapidly under these conditions and come to dominate the spoilage microflora that develop. The first indication of the spoilage in fresh meat is the production of odors. Bacterial metabolism produces a complex mixture of the volatile ester, alcohol, ketones, and sulfur containing compound, which is collectively compromise the off odor. Studies have confirmed the predominant role of the Pseudomonas in spoilage of the aerobically stored chilled meat. In the later stage of this spoilage, which can be increases in a meat pH is seen when a microbial number are reach higher level. In further indication of this spoilage, it become apparent in the form of visible surface slime on the meat. Then we can use a packaging material, that is your vacuum and modified atmospheric packaging of the meat, which changes the microflora of the meat and it constantly time, course, and character of the spoilage. Meat can be processed in a number of different ways, which can affect its characteristic shelf life and microbiology. These diagrams show that. Okay, so freezing at low temperature, canned milk, modified atmospheric packet, vacuum packaging, fermented milk, pH, and it is your cured milk. Now, after meat, the next is your plant products. So, plant product also contribute a greater part for a human consumption. We all know that every part of the plant structure are used from root, tuber, stem, leaves, fruit, and seeds for the consumption of the human being. Now see what is written here. The plant kingdom provide a considerable part of the human food requirement. We all know that. Okay. Apart from the meat, the vegetarian population get get the nutrient from the plant kingdom. So these plant kingdom play a very vital role for a requirement of the food in case of the human. Okay, we all know that every part of the plant structure, like either it is root, tuber, stem, leaf, fruit, and seed, they are consumable. Plant produce product are grow at large scale and many are stored for a world to another. Microbiological problem may occur at all the stages in the production of the plant products. So, if the plant are used for the consumption, then obviously the microbiological problems are also occur, and this microbiological problems are occur at all stages when a plant product are produced. During growth, there is a wide range of plant pathogens to contend with, and although these are dominated by the fungi, there are significant number of the bacteria and the viruses. Plants have many mechanisms to prevent the microbial invasion of their tissues. The outer surface plant tissue may contain the antimicrobial agent, and the low pH in case of the fruits and high pH in case of the vegetable, and availability of the water. They are what? They contributed the microbial phenomena. These three groups of the plant product, that is your fruits, vegetable, and cereal, they have a sufficiently distinct that they will they will now be considered separately. The first one is your cereal. Okay, some of the more important cereal crops like your wheat, rice, and corn. So we can talk about all these products. That is your cereal. So the microbiology of cereal during the growth harvest is dominant by the mold. Okay. So when a cereal grow and harvested, so it can be spoiled by the mold. Although they required a relatively high water activity for the optimum growth, like Cladosporium alternaria. The most important genera of storage fungi are Penicillium, Aspergillus, although these species of the Fusarium may also be involved in spoilage when the grain is stored under the moist condition. When we store the wheat at grain at moist condition, then there is a chances of the growth of the other fungi also, that is your Penicillium, Aspergillus, and your Fusarium. Water activity and temperature are the most important environment factors which influence the growth of the mold in a cereal and the possible production of the mycotoxin. When the grow in a cereal, then it contribute into the mycotoxins. Xerophilic mold like your Urotoium species and Aspergillus restrictors may grow very slowly at the lower limit of their water activity Once they start growing and metabolizing, they will produce water of respiration, and the local water activity will steadily rise, allowing more rapid growth. There is a sequence of observable consequences of the process of the mold growth on cereal, starting with a decrease in germinability of the grain, which is followed by the discoloration, production of the mold metabolites, including the mycotoxin. Demonstrable increase in temperature, self-heating, the production of musty odor, caking, and a rapid increase in water activity, which leads to the complete decay with the growth of wide range of the microorganisms. Now, preservation of high moisture cereal, like suppose that if we have a high moisture cereal like your barley, so we know this barley also provide a high nutritional winter feed for the cattle. Long term storage of it can be achieved by a lactic acid of fermentation or by the carefully addition of the fatty acid, that is your propanoic acid. Propanoic acid inhibit the normal spoilage mold which are associated with this cereal in a temperature climate, but not enough to inhibit the aspergillus flavus. This mold can produce a alpha toxin B1 under these conditions if such material is fed to a dairy cattle. Alpha toxin M1 being will be secreted in the milk, which cause a problem in human food with a problem of animal feed. Preservation of pulses, nuts, and oil seed. The pulses are the major source of the vegetable protein. We all know that. Okay, so if someone are vegetarian, then they get their protein from the pulses, which included the important crops like your peas, beans, soya, groundnut, and lentils. The mature seeds of the soya bean and the groundnut are harvested. Longer term storage and maybe spoilage with mold if not stored under the appropriate condition. Several of the groundnuts and soya bean sources of the vegetable oil. Edible nut which rich in oil like your groundnut have a much lower water content at a particular water activity than the cereals. Preservation of the cereals, nut and oil seeds. Pulses and oilseed. Apart from the problem of the mycotoxin, there is a formation of the molded oil seeds. Several mold species have a strong lipolytic activity. We all know that there are several mold species which have a strong lipolytic activities, which leads to the contamination of the extracted oil with free fatty acid, with may in turn undergo to the oxidation to form a product which contribute to the rancidity. The most important type of the lipolytic molds are Aspergillus niger, Penicillium, while at higher water activity, species of the rhizosomes are also important. If the cereal, pulses, oil seed and the tree nuts are harvested, then there is a little damage occur. And dry to an appropriate water contain it till store at considerable period of time, so long as they are not exposed. Excessive temperature abuse during the storage. The problem which may arise when the large storage facilities like your silos, they are not carefully designed to avoid the temperature differential arising within a store community. The migration of the water in these circumstances can result in a germination of the fungal spores and the growth of the mycelium. Ravage of the pests such as an insect and rodents and their activities. Fruits and fruit product. The degree of these specificities are shown by many of the species of the mold active in a spoilage of the harvested fruits in the marketplace or the domestic fruit bowl. Okay, so these fruits can be spoiled either during the harvesting or during the marketing place or during the domestic fruit bowl, and it can be um, spoiled by the mold, reflect their possible roles or as pathogens or endophytes of the plant before the harvest. Does the Penicillium italicum and Penicillium digitatum show a considerable specificity for the fruit, that is your citrus fruit, being the blue mold and the green mold respectively? It can damage the orange, lemon, and other citrus fruits. Penicillium expenses cause a soft root of apples. These species cause a blue mold of the apple. This particularly species has a ability to produce the mycotoxin pathogen as a contaminant in apple juices. Other common disease of apple and pears, which included the black spot or scab, which caused by the Ascomycete venturia inaculis. A brown root are caused by another type of the species, that is your Ascomycetes monilinia fructigina, but it does not causes any extensive rooting of the tissue. And especially widespread mold on both type of the fruits and vegetable is a gray mold. Okay, that is your botrytis cinerea, which is the impact imperfect stage of another Ascomycetes, Botania fucaliana. Moldy fruits during the storage and transport should be removed and destroyed. Good hygiene of container and packaging equipment is essential to prevent buildup of mold propagation. Some biocide was used to prevent the mold spoilage. Benomyl has proven useful where it can be applied to the surface of the fruit like your citrus and banana, in which the skin would normally be discarded. Penicillium digitatum have developed increased the resistance to the benomyl. Biphenyl is quite an effective protectant when incorporated into the wrapping tissue of the fruit like your oranges when they are individually wrapped. Captain has been used as a spray for the strawberry fruit, but its use must be stopped well before the harvested. Reduce temperature and increase the concentration of the carbon dioxide also control the mold spoilage. Okay, if we store the fruits under the low temperature and if we increase the carbon dioxide concentration, then they both also control the mold spoilage during this storage and transport. But many fruits are themselves sensitive to a low temperature and enhance of the carbon dioxide level. Canned fruits are normally useful in controlling the mold spoilage during the storage and transport. Vegetables and vegetable products. Now, the cereal are completed. The fruits and their fruits products are completed. The last is your vegetable and vegetable products. Okay, if the higher pH value of the tissue of the many vegetable make them more affected by the bacterial invasion than the fruit. If the pH value of the vegetable tissue are higher, then it can more affected by the bacterial invasion as compared to the fruits. The bacteria involved are usually pectinolytic species of the gram-negative genera, that is your Irvenaria, Pseudomonas, and Xanthomonas, Clostridium, and the non-sporing gram-positive microorganism, that is your Cronidibacterium cipidoni. However, the most frequent agent of the However, the most frequent agent of spoilage are not the plant pathogen themselves, but opportunity of the microorganism which gain access to the plant tissue through our wounds, cracks, insect damage, or even the lesion which is caused by the plant pathogens. All freshly harvested vegetable have a natural surface flora, including the low number of the pacnolactic bacteria, and it's become intrinsically evident that the healthy tissue of the intact plant may also contain very low number of viable microorganisms, that is your endophytic. The onset and rate of spoilage will depend upon the interaction between the physiological changes occurring in the tissue after harvest and changes in the microbial activity. Harvesting itself will produce physiological stress, principally as a result of water loss, wilting, and cut surfaces may release the nutrient from the microbial growth. This stress may also allow the growth of the otherwise quiescent endophytic flora. The most frequently the most frequently observed from the spoilage is softening of the tissue, which occur due to the tactolytic activity of the microorganisms. We all know that actin is a major component between the cell making up plant tissue. One is broken down, the tissue loses its integrity. When the packing is loosened down, the the tissue loses its integrity and the individual plant cell are more easily invaded and killed. The control of the relatively humidity and the composition of the atmosphere in which the vegetable are stored is also play a vital role for the contamination reduction. Like relatively humidity, because its value is at the range of 90 to 95%. Loss of water from the vegetable tissue will lead to the wilting. It is essential to avoid the presence of free water on the surface of the vegetable, and the temperature control may be just an important prevent condensation. The presence of the water on the surface will allow access of motile bacteria like your Erwinia and Pseudomonads to wounds. Vegetables should not normally be cause of public health concern, but transmission of the pathogenic bacteria like your Salmonella, Shigella. It is a possible by direct contamination from the fecus of the bird and animal. The use of the sewage sludge as fertilizer or the use of the contaminated waters for the irrigation. Not all the pathogens are transmitted to the vegetable by a direct or indirect fecal contamination. Like your Clostridium botulinum, it can be contaminate with the spores of the microorganisms. Okay, another type of the pathogen which are naturally present into the environment, like your psychotrophic species, that is your Listeria monocytogenes, which is commonly associated with the plant material, soil, animal, sewage, and a wide range of other environmental sources. The two other psychotrophic microorganisms, that is your Yersinia enterocolitica and Aeromonas hydrophilia. Both may be expected to be associated with the vegetable, which can cause a illness. Okay, so this is all about the food spoilage and the different food that we already discussed. That is your milk, meat, cereal, vegetables, and fruits, which is a major source of our diet, and how it can be spoiled and how we prevent the spoilage of these type of the food products. Okay, so this is all about the food spoilage. I hope you will understand, but if you have any query related to this, you are freely to ask me in a comment box, or For a further detail, you can follow the ITLS Academy on different platforms. That is your Instagram, Facebook, Twitter, YouTube, WhatsApp, and LinkedIn. And subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. You can also ask your query to the helpline number that are already mentioned in your slide. That is 7080833450, and visit our website, that is www.itlsacademy.com. Thank you for watching. Please like, share, and subscribe our channel.